It caters to a unique characteristic of each patient, and this kind of treatment is very promising for the future, so that it is going to take care for the patient's needs, in the sense that every person is unique in the genetic architecture, and hopefully it will be a very interesting session for all of us over here. We have very honored dignitaries who are here as chairs, and they are the best fit for the session. So I'll give a quick introduction. Our first chair is Dr. Madhusudan R. Nandineni. He is a staff scientist and group leader at Center for DNA Fingerprinting and Diagnostics. Dr. Nandinini's current research interests include forensic DNA profiling, human population diversity, and plant-fungal interactions. Recognized with accolades with as Innovative Young Biotechnologist Award and Max Planck India Visiting Scientist Fellowship. He is elected fellow and executive council member of the Telangana Academy of Sciences and a member of the board of trustees for the Max Planck Institute for Plant Breeding Research, Germany. His group's primary focus is exploring genetic diversity within Indian populations and conduct genotype-phenotype correlation studies, validating SNPs associated with visible traits such as skin color, BMI, and height. Furthermore, they investigate pathogenicity genes in Colectotrichum truncatum, aiming to understand its infection biology, lifestyle, and host specificity in plant microbe interaction. A huge round of applause for Dr. Madhusudan R. Nandineni. Our next chair is Dr. Kiran K. Delukunta, who is currently working as a computing systems development worker at Institute of Organic Chemistry and Biochemistry of the Czech Academy of Sciences, Prague, to maintain and develop HPC cluster infrastructure for bioinformatics computations. He endorses the application of renewable energy and works part-time as Information Systems Administrator in International Solar Energy Society, Germany. He is equipped with strong information technology skills and promotes the usage of the Galaxy framework. He believes in the consciousness of science and technology for the welfare of humanity and contributing to the world through India. He is one of the directors of the TMS Foundation. In 2018, he completed his doctoral research in pharmaceutical bioinformatics at University of Freiburg, while managing several scientific IT infrastructure for bioinformatics and cheminformatics scientific projects under Professor Stephen Gunther. Over to you, sir. A huge round of applause for Dr. Kiran. So our first speaker does not need an introduction, but I will pass over the mic to the chairs to introduce him.
Yeah, thank you. Thank you for the nice introduction. It is my great pleasure to introduce our distinguished speaker for this session, Professor Thangaraj, a renowned scientist and the former director of Center for DNA Fingerprinting and Diagnostics. So I don't think there is anyone in this hall, almost like 90% of the people here are associated with Dr. Thangaraj work some way or the other. And I mean, the research had a profound impact in the field of human genetics, health, and disease. This group has made significant strides in understanding the complex origin and activities of Indian population. This research also explores the ethical implications of endogamy, which is the practice of marrying within the specific group, on the health and disease patterns of the Asian population. In addition, Dr. Thangaraj's work extends to uncovering the molecular foundations of important conditions, including male fertility, cardiovascular diseases, sex discrimination, and mitochondrial disorders. Dr. Thangaraj will today be sharing his insights on population genomics for public health. His talk will highlight the significant role that population genomics can play in improving public health strategies, especially in understanding genetic predispositions to diseases and informing better healthcare policies. Dr. Thangaraj, with this, I'll request you to please deliver your talk. There is no timing here. Whenever you stop, we will stop.
Thank you for generous introduction, also for generously giving me time, but of course I'll try to finish within half an hour to maybe 35 minutes. Thank you very much for the kind introduction. Also thank Naneshwar and his Jnaneshwar and his team for organizing AdNet meeting and bringing people around the world who are doing extremely good science. Most importantly, personally, I feel because he brought a lot of people who are associated with CCMB, that gives me a lot of pleasure to meet most of them whom I have not seen for months or years. So what I'm going to talk to you is about how studying the population genetics or population genomics is helping us for better understanding of our health and disease, particularly understanding the rare diseases. When you talk about population genetics or population genomics, there is no other place in the world which has such a huge diversity. As you see in the picture, we have people who are similar to Africans, people who are similar to European, people who are similar to Southeast Asian, and there are people who are very unique to India. Okay, so that slide summarizes all this. Over a period of 3 decades, we have been trying to understand why such a huge complexity in terms of population histories exists in India. And I'm not going to talk all of these, and it's my pleasure to tell you that most of the people who have contributed to this are here. I don't want to take a name. I am afraid that I may leave a lot of names, because at least 30 to 40 people, not only Indians, also international scientists are associated with these studies. From this, I'm going to take 1 or 2 examples how these have helped us in taking the study further to help the common people. Okay, so I'm going to take you 1 or 2 studies. In this particular study, what we have actually shown was that in the prehistoric India, there were 2 founding populations. One, we call them as ancestral South Indian, who are probably part of the early human migration. As you know, that Andamanis were the first modern human who migrated out of Africa, which we have demonstrated long ago, 2005, where Jnaneshwar and others are associated with that. There was another wave of migration which brought people to north, and we call them as ancestral North Indians, who are also closely related to Europeans. Again, there are several other studies have shown that how genetically these 2 groups are connected, which I'm not going to detail. On the other side, if you look at the PCA plot, where we have a lot of Indians here, this is a European HapMap sample, and these are African HapMap samples, and these are the Chinese HapMap samples. While you see that very strong clusters where hundreds of individuals, suggesting that genetically they are more homogeneous, whereas Indians, every single population is very unique. That means India is not inhabited by one single large population, whereas you can say India is inhabited by large number of small populations. And these 2 founding groups have admixed and gave rise to many populations. That again you can see in the PCA plot, that although every single population is unique, but there are some connection between the groups. That's why they form a gradient of cluster. So later, at this point of time, we don't know when this admixture took place. So we took another study to understand when this admixture between ancestral South Indian and ancestral North Indians have taken place. Here what we have shown, these 2 founding groups actually gave rise to many populations subsequently, from one after the other. Maybe you can imagine that the initial population, the founding population, settled in some jungle in southern part of India, either Kerala or Tamil Nadu. From there, one group must have diverged from there. Probably they finish all the food available in the forest, they must have migrated to other place, and from there to other place. So that's how the population must have emerged on both the side, ancestral South Indians and ancestral North Indians. These two groups have admixed during the last 2 to 4,000 years, genetically mixed. That's what we are seeing now in the PCA plot, that every single population is connected because of the admixture. They all share some kind of genetic affinities. Then after admixture, for the last 2,000 years, every single population started maintaining the endogamy, means marrying within the group, right? So this is what kind of summary that what is the population exists today. Initially, two founding populations gave rise to many populations. There was admixture, and now every single population for the last 2,000 years maintaining the endogamy, right? And probably the caste system which came later on must have put more pressure to strictly maintaining the endogamy. Even today, we see that most of the populations in India are maintaining the endogamy. Now, I'm going to take you to that. So, what is the impact of the endogamy, which every single population marrying within the group, on our health and disease? So, now to understand, we are not restricting only with India. Now we wanted to take it to South Asia because of several reasons. So one of the reasons in terms of health perspective, this is our earlier study. What we have shown that there's a mutation in a gene called myosin binding protein C3. There's a novel 25 base pair deletion, which actually lead to three different kind of cardiomyopathies: dilated, hypertrophy, and restrictive. So all of them suddenly it leads to sudden cardiac arrest. When the mutation exists in the homozygous condition, the individual may not survive beyond 10 years, which actually we estimated by looking at the postmortem tissue of the children who have died at very, very early age. But the individual who carry the heterozygous mutation can live up to 40 or 45 years. Then it shows symptoms and then it leads to sudden cardiac arrest. So that mutation is restricted only in South Asia, but not in other countries. Therefore, so whatever we study in terms of genetics, in terms of disease in India is also relevant to other South Asian countries. So therefore, we took a large number of samples, 2,800 individuals from 275 endogamous group. As you see that these are the place where the samples have been collected, which includes the four major linguistic groups: Dravidian, Indo-European, Austroasiatic, Tibeto-Burman. Also, there are some isolated languages like Andamanis, Nepalese, and few more. So this study, what we have actually found was that it's not really interesting to come to know, but what we have found is that there's a lot of opportunity for us to identify a population-specific recessive mutation which might lead to population-specific recessive disease. The reason why I'm saying this is that if you look at the IBD score, identity by descent, there are two populations here which is highlighted in red color. One is Ashkenazi Jewish and the other one is Finnish population. Those two have tiny IBD, but still they have large number of diseases. Mostly they're all recessive disease, except few dominant. And these are the diseases and these are the genes which are having the mutation. And these are Finnish population, again large number of diseases. So why I am highlighting the recessive disease is that if the dominant disease, as all of you are aware, in a heterozygous condition, it will lead to a disease. So whosoever carry the mutation in one of the chromosome, they will certainly have the disease. Therefore, obviously, no individual carrying the mutation looks normal. Whereas in case of recessive disease, those who carry the mutation heterozygous condition apparently look normal, but in a society like India where the endogamy is practiced, marrying within the group, if there is some founding mutation, it is a heterozygous recessive. The individual who carry either male or female absolutely will be fine, but when he or she can transmit that to the next generation, again next generation will be fine. But if it goes to generation after generation, after few generation, because the people are marrying within the group, there's a possibility that two individuals carrying the mutation in heterozygous condition, if they marry, and there's a possibility that they contribute the abnormal chromosome to the next generation, both father and mother, the child receiving the homozygous condition will have the disease. So that is the reason that why India having large number of recessive disease, particularly if you take rare disease, there are 70 million people are affected with the rare disease. So in that condition, what I have given you, if you look at the pedigree, then you can able to understand, for particularly for the students. This is a pedigree where this lady, for example, carrying the mutation heterozygous condition, she will be absolutely fine. She married someone who doesn't have the mutation, then the mutation is transmitted to the next generation. Two of their daughter carry the mutation. Again, they married somebody from outside, therefore no problem in this generation, again this generation. But now here, or if you take furthermore generations, both this girl and boy carry the mutation, heterozygous condition, they will be absolutely fine. But when they marry, there's a possibility, 25% of possibility that they can transmit this abnormal chromosome to the next generation. Therefore, there's a disease. That condition is quite high, which I will highlight in the coming slides. So having this, you can imagine that with this tiny IBD itself, which I showed, there are large number of disease, maybe 25, 30 disease has been mapped. But what would be the consequences of large number of population in India? So every single line represent one population. Okay, so most of the population have several fold higher IBD. That means in a given population, if you take few individual, they will have the same haplotype because they have inherited those from their founder, right? So to understand, really, when we collected this sample, we don't have any clue at all whether the population carrying any disease or not. So we went back by identifying, is there any disease exists in the population or not. So we selected few population from southern part of India. So we went back, try to understand, is there any such disease, recessive disease particularly exists in the population. Then we came to know that there's a disease called junctional erythritidis epidermolysis bullosa in a one particular population, southern Tamil Nadu near Madurai. That there's a disease which has skin rashes and congenital heart disease. Unfortunately, the children at the time they're normal, but immediately they develop the disease, and within 2 months of the age they die. Such a very, very sad situation. But if you go to the population in a given time, you don't see the disease because the children with the disease they die, right? But heterozygous individual parents, absolutely normal, they live without any problem. Then we screened for this mutation, initially the known mutation, we could not find anything. Then we did exome sequencing and found 11 base pair novel deletion in a gene called LAMB3. Right? So then we had an opportunity to collect more sample for this Genome India project, which sequenced 10,000 individuals, which will come a bit later. So we had a camp in the place where we collected samples and screened. We collected about 150 samples, and we found 10 of them in that, maybe up to 10% or little more than that, carrying this mutation. When we did the haplotype analysis, we found this is a wild type chromosome, this is with the mutant, and you can see this chromosome differ, whereas the chromosome which carry the deletion, 11 base pair deletion, is constant in all of them, suggesting that there's a founder mutation few generation back, now it is well spread across the population, right? Then we went on to see how it is distributed within India with our own samples. And we also took samples from the 10,000 genome which we sequenced that actually represent all over India. We collected 20,000 samples. And in fact, this 10,000 genome has just been completed, and this was a announcement of completion of the 10,000 genome by the Minister of Science and Technology, the presence of Secretary DBT. So I'm one of the coordinators coordinating the program, and all of these 10,000 are in IBDC, Indian Biological Data Center, and we are actually trying to release it by January 2025. DBT is drafting the guidelines and so on. Hopefully, it will be available for everybody, every researcher to make use of that. So this is to tell you that when we sequence, when we look for the mutation, the 10,000 genome, we could not find any of them carrying this mutation. Then we also screen for this mutation in a global database. These are all different databases. Screened in 8,62,000. If you take the 10,000, 72,000, except two individuals from Genome AD, that also from South Asia. So none of them have this mutation. That means it is so rare, rarest of the rare disease. But when you go to the population, it is highly prevalent. That's because of the endogamy and founder mutation, right? So we don't have any clue about the functional aspect of this gene or this mutation. So we created animal model using CRISPR-Cas, and we developed the homozygous 11 base pair deletion. So in this case, what happened? This is a wild type. This is with the mutant. The skin is wrinkled, and there's a blisters coming within 1 day. Then the next day, animal dies. But the human survive 2 months, where the pups are dying within 2 days, right? And this again is not allowing us to take it further to understanding the biology or the functional aspect of that. But fortunately, during this process, we got one more clone with 45 base pair deletion that includes that 11 base pair. This is non-frame shift. So we proceeded with this to see what is happening there. In this 45 base pair deletion animal, the symptoms start appearing after the 4 months, as you see here. Typically, after 4.5 months, and after 6 months, you can see the animal looks like this. Typically, the phenotype which we have seen in the patient. And the internal organs, the previous slide which I showed, its inflammation, and here the heart is enlarged, spleen also enlarged, but stomach is become very small. So in this case, we don't know what is actually happening in those tissues. So we are right now doing a micro CT to understand the structure of this. Then we also looking at the skin. What is difference between the wild type and the mutant? In the wild type, you can see epidermis and dermis is nicely organized, very compressed, whereas in the 45 base pair deletion animal, this is very loose and also very thick, right? And there's a gap between epidermis and dermis. This is where the blister starts appearing, right? So that gives kind of clue that what is really happening on the skin. Then one more interesting thing which we found was actually the 45 base pair deletion happening in the splice site, right? So when we isolated mRNA, converted into cDNA and looked at, then we can actually see there is a splicing out of exon 21. You can say exon 20, exon 22. This exon 22 is actually spliced out. So therefore, probably it has variable kind of phenotype, and the late onset of these phenotypes are happening in this case. Otherwise, this is a non-frame shift mutation. Because of splicing, these things are happening. Until now, we looked at the population level, we looked at what is really happening there. But now, as a responsible citizen, a responsible scientist, whether can we do something for the family? We came to know that one mother was pregnant. Then there's no facility in Madurai, close to Madurai. Then we brought the mother to Chennai, collected the amniotic fluid. Then we try to do just simple PCR based method to find out whether the fetus carrying heterozygous mutation or homozygous mutation. Then we found this is mother's heterozygous and this is also heterozygous. We confirmed that by Sanger sequencing, and you can see that multiple peak suggesting that the fetus also carrying the heterozygous mutation. Therefore, we advised the mother to continue with the pregnancy. And the child born 23 June with the normal phenotype, that gives a kind of satisfaction for us that whatever we have done for the last 3 decades and now coming to help of the population. Then we also wanted to create the homozygous line in human because the children are dying at very early age. Only heterozygous we can get from the parent. We use the amniotic fluid, which we use for diagnosis, that's carrying the heterozygous mutation, and we actually generated iPSCs in Tata Institute for Genetics and Society with the help of Vasant, and actually he used the pluripotency marker as well as trilineage marker to see whether it has a potential to transform into different lineages. And this process, he successfully created the homozygous line with the deletion, and these are positive. So this is where we are now. We are going to use it further to understand, develop into skin lineage and so on, to look at what is actually is happening in this case. Having all this information, because again one step we'll have to go further, we are now approaching Tamil Nadu government to give us the permission to go to the village to screen entire population. Although this particular community is living in 5 different districts, but this mutation is in only one district, right? So the population is common, but what is happening here, there's a founder mutation that is circulating only within that village. Even they are not going out of the village and marrying others from the same community. So much structural things happening within that village, right? So there are only 5,000 individuals. We wanted to screen all of them, and using those information, we wanted to give premarital and prenatal counseling, right? So that if we continue this for next few generations with the help of the public health officials, I would say that Tamil Nadu government, I don't know about other state, they have very good public health system in very rural. I myself experienced in very midnight, the people are very helpful, right? So if you train them every pregnancy or before marriage, premarital counseling, we can actually eliminate this mutation after a few years or few generations. So that is what is our interest now, and if this is successful, and we are actually, there are 4 different stages. So the Tamil Nadu Medical University, MGR Medical University, is attached with this public health department. We have to get their approval. Initially, their scientific committee approval, then ethical committee, those are over. Now it is the process of public health department. If they give, we'll go and do all of those. So if it is successful, we want to replicate. We also identify similar condition in few other states. So we wanted to extend that to the next state. In this connection or continuation of that, because we are knowing such a deep recessive disease exists in India, rare disease exists in India. When I was heading Center for DNA Fingerprinting and Diagnostics, so I initiated a program called Mission Program on Pediatric Rare Genetic Disorders. There are 16 laboratories, as you see the dots, are part of it. BHU is part of it. RGCB Moenak is part of it, and many more investigators are part of this program. One good aspect is that all of them are routinely doing the genetic screening in their own institute. Now, if they identify any unique case of rare disease, if they are not actually known to have any of the known mutation, then the sample will come to CDFD. CDFD will do exome sequencing, and we also have telemedicine facility. We established one full wing for this, and we do routine in silico analysis, and if there is a mutation which potentially might cause the disease, then we'll use one of these model system to understand, as I mentioned, the mouse model. Then take it to the patient, counseling and so on, and we also try to do some kind of gene therapy using antiviral based method. And most importantly, this is an awareness program. The small videos of 8 different languages for public awareness. If somebody is really interested, you can go to this particular website and see. You can see all the participating institute. You can also see the videos in 8 different languages, which will be useful for the society. So this actually was, this lab has been inaugurated by the then Vice President Sri Venkaiah Naidu, and this program was launched by Dr. Rajesh Gokhale, Secretary DBT. So this is going to be one of the very, very important program for years to come. So it's a 5-year program. And with that, I'll stop by thanking all of those who have contributed. Again, very long list, right from our 3 decades of work, including Dr. Singh and many others. Deepak and Karthika, the one who have contributed a lot to the recent mouse model study, and list goes. And the funding has come from CSIR, UK India Education Research Initiative Grant, DBT, DST, and Medical Research Council London, which is ongoing project. And thank you very much.
Thank you, Thangaraj, for the excellent work. And this talk actually exemplifies how work from a population genetics variation, you take it to a disease model, then you look into the population. It actually is a textbook kind of example how you can translate the research from basic research to societally relevant research. That Rajesh always tell us whenever the student at CCMB. Societally relevant research. And we have time for 2 quick questions. If anybody has questions, please. 2 quick questions.
Yes, Professor Rao.
Professor Rao and 2 questions.
Congratulations, Thangaraj. See, Indian population structure, consanguinity and heredity. We know that the south more consanguinity, north west no consanguinity. From that perspective, you are out of genomics data. You are rightly addressing the issue of genomics. to rare diseases, where genetics can definitely help the people. The question here is, what could be the functionality and practice? We know 65,000 years of existence, but what do you expect from what based on the genomics? What could be the age of this practice of consanguinity? Any insights?
So we estimated the age of the population-specific marriage, endogamy, that happened 1900 to 2000 years. But the consanguinity probably much older than this, because when there is a few families, they should have married within that. Then it is expanded, then become endogamy.
Something to do with the breeding. I mean, basically
Yes,
it's the breeding
correct.
because the population was small and then they were isolated.
That's right.
So, the founder effect is when there's loss of genetic variation when any population species.
That
So
mic
the
is not
founder
on.
effect. So the founder effect is when there's loss of speciation when two populations diverge, and the LAMB3 gene, there's a close parallel of LAMB2 gene. So is it because of the parallel? Can you say that that's a large common ancestor for both the ANI and ASI?
No, we cannot actually say that. If it is a the mutation is very old, right? Then you expect the population, most of the individuals of the population carry that. If they're very old and recessive in nature, right? And also it's not actually giving disadvantage to the population, not only the disease or any other condition. Otherwise it will die automatically. If it is not really disadvantage in the recessive condition, very old, then you expect it is spread across the many population. But if the mutation is very recent, like the LAMB3, happened after the divergence of populations. Even the same population that is restricted to one particular village, that means it must have happened very recent. Unlike the myosin binding protein C3 gene, the mutation we predicted, it happened 33,000 years back, okay, before the ANI ASI admixed. Therefore, it actually is there across India and across South Asia. So all depending upon when this mutation arose and when how it actually spread can be established. Whether that was your
And the quick question, just like a pan-genome, can you extrapolate it to a super reference genome where every node is a base and along the traversal of the quad tree, we can essentially accommodate all reference genomes there. So, like a pan-genome is for species, a super reference genome can be across species with quad trees such that every node, there's a divergence of 4 nodes, single basis, and from traversal from node to leaves, more is traversing towards the leaves, depending on sequence similarity, we can have closely related reference genomes of species.
no country like India, we cannot have super reference genome because every single population is very unique, and just I can tell you that there are millions of variation between the two different population in India. That's what giving just come from the 10,000 genome. So in that scenario, you cannot actually have a super reference genome, right? Unless otherwise, there's no bar between the population, and every population across India are marrying, right? So the situation like in India, like endogamy, we cannot actually have that kind of thing.
So, super reference genome,
So we can continue this discussion. So Thangaraj, I just have a small question. Has somebody looked at the protein because the 15-day amino acid deletion has less severe effect than 11 days to definitely not of pain. Has somebody looked at the protein?
Yeah, we are doing proteomics now. There are many things are going on which I could not.
There are no questions. I would like to thank Dr. Thangaraj for his excellent talk. You have taken us from ANSI to ASI to population genetics to translation research and to societally relevant research. Thank you very much for your talk. Please give him a now we will have
Thank you.
speaker, Dr. Kiran.
So now we are honored to introduce our second speaker, whose work is greatly impacted the field of neurodegenerative diseases, specifically amyotrophic sclerosis, also known as love-giving disease. Next speaker is none other than Professor Udaykant Pandey, a renowned professor at the University of Pittsburgh Medical Center, Pennsylvania. Professor Pandey research focuses on unraveling the molecular mechanism of ALS and other related motor neuron diseases. His work is at the forefront of understanding genetic and molecular underpinnings of these devastating conditions. One of the most exciting aspects of Dr. Pandey research is his investigation